great to be back. I thought about putting on a bow tie because my job is to play Bill Nye Science Guy for you guys. I decided not to. I decided to make it fishing edition, by the way, because we're going to talk about fishing, mainly looking for genes first. So what I want to go over this morning, and some of it will be a little bit overlapping, hopefully in a good way, with some of the talks, the wonderful talks we just heard in the first session, but I want to talk a little bit about finding genes for CFC, because we still have a little bit of ways to go on that. I want to talk about what I call the mouse catwalk and other unlikely tales, and then about getting better, and then very quickly at the end I want to say a few words about the ClinGen registry, which is a follow-up to the talk we heard about the registry that you all have developed. So you heard in a very beautiful way from Kate about how the initial genes for CFC were found. They were found initially because of overlap because of Noonan syndrome, and we knew that Noonan syndrome was in this thing, the RAS pathway. And so our first idea actually was the notion that the Noonan gene would cause CFC, and of course that didn't work out. And that then led to the idea that if we look at genes that, since the two overlap, that genes are similar in a similar pathway, that we'll get there. Of course, as Kate explained very nicely, that has led to the discoveries we now use. And here's the pathway. So, and the problem we face at the moment is that although most of you, I know from yesterday, will have had gene testing done and will have been successful in identifying the gene that underlies your child's CFC, there are a small percentage of individuals, something on the order of 20% of cases, where that same gene test is sent off and it comes back negative, and yet the doctors are clear that this child has CFC. So the question is, what's wrong? What are we missing? And most importantly, how are we going to fill in the missing 20%? So the way we found the genes so far, and these are shown on this as a not complete diagram. It shows not just the genes for CFC, but the genes for the other traits like Noonan syndrome, Costello syndrome, and so on. You can see that they are spread all over the pathway. And the way they were found was by just checking all the genes in the pathway and seeing if they had mutations in kids who had these diseases. But at a certain point, we've run out of ideas, actually, in terms of resequencing these genes, and yet we still have kids who are unexplained. So how do we move forward? And the answer to that comes through some pretty amazing advances in DNA sequencing technology. And actually, this is what underlies the testing that many of you have already had, which is so-called panel testing. So some of you will remember the day back in early 2000, 2001, where there was a ceremony in the White House, outside the White House in the Rose Garden, and Francis Collins shook hands, and they announced that the human genome had been sequenced. So that was an amazing thing, and if we had decided the day after that we wanted to sequence one of our genomes, we could have then done so for the mere cost of about $100 million. That does not make for very good genetic testing, of course, and fortunately what has happened, that's what this graph is showing, is the price for sequencing genome has dropped in a way that's pretty amazing. Shown in the line, is Moore's law. That's the rate at which pricing has come down for computing, which is a pretty amazing thing in and of itself. And you will see that the price of DNA sequencing has crushed that, right? So as of today, it costs only on the order, just for the DNA sequencing part, the actual test costs a whole lot more, but it only costs about $1,500 to sequence someone's genome, so from about $100 million to $1,500, that's a pretty amazing multiple-fold reduction in cost. And sometimes we choose not to do that because we don't need to know the whole genome, and we just want to sequence the part that has the instructions, the so-called exome, and we can now do that for about $350. So that has really opened up possibilities for research. So I would imagine the way we are going to figure out the remaining genes that we don't know about yet for CFC is have it out on a safari. And for the most part, the way we will do that is by taking individuals who have CFC and their parents who do not have CFC, and then sequencing at least the part of the genome. It's about 2.5 million base pairs. It can be done in about 2 weeks. and look for the DNA changes in that affected child that are not there in the parents. And in fact, some of you, I know from yesterday, some of you are here already had this sort of approach. The difference is, instead of looking at the genes we already know about, we can also start to look for genes we don't know about. And just so you know, if we looked at any of us, your children, us, whoever, we only find about 1 to 2 serious changes per exon, ones that actually change the DNA instruction. So it's not a whole lot of sorting out to find it. Now, it's not a lock-clad guarantee that we find genes, but this is the way that we're going to close the gap between a test that currently accounts for about 80% to a test that can maybe account for all cases of CFC. Okay. On to the catalog. So obviously catalog refers to modeling, so I'm going to tell you a bit of that using animal models to study CFC. And so far, most of the animal models have been done with 2 organisms. On the left is the very overgrown version of a fruit fly. You will know it as the little tiny annoying things that fly around the bananas sometimes in the supermarket or such. And then, of course, there's the model of mice. And the reason this particular mouse is shown geared up for a boxing match is because we can both knock out or knock in changes to mouse genomes to make models. So we can actually put in the changes that cause disorders like CFC once we know what they are. So why do we use animal models in the first place? And here are some ideas about how these models are currently being used. One question, or a set of questions, is around the issue of exactly how does CFC happen. Of course, we know the gene change, but that doesn't tell us why is it that we have the skin changes, or the seizures, or the heart disease, and so on. And so we're able to ask questions like, why are certain organs affected? And on the other hand, because it turns out that the RAS pathway is important sort of head to toe, why is it that certain parts of the body are spared, unaffected in CFC? We don't know that. And then, and this was something that Kate alluded to, we know very well that even with the very same mutation, that there can be differences, certainly different mutations, there are differences in CFC amongst individuals, and animal models are a very strong way to begin to understand how much of that is the other genes that she referred to, or maybe the environment and so on. So we can do that. And the other tremendous use for these animal models is looking at treatments. It's obviously preferable to have some confidence before we start trying things in patients, your children, that it might work and that it wouldn't be dangerous. And so these are the kinds of things we can address. So which particular treatments would be used and when in life would we be giving these exactly. So I'm going to tell you a little bit about first the fly models and then I'm going to tell you about the mouse models. And I'm going to focus for the fly models because there have been 2 papers, really good papers, in the last year that have specifically looked at, used fly models to look at CFC. It's the work of a colleague of mine, I'm working with him, Stas Schwartzman, who is at Princeton and will be at the science meeting in Orlando at the end of next month. He chose to use MEC1 to model, and you might ask, why MEC1? Because BRAF is so much more common in CFC, and the reason is because MEC is biologically simple. It only targets and only signals down to one protein, namely MAP kinase right below it. So it's the simplest of the proteins, and so that's why he chose this. And to give you an idea of the kinds of things you can do with a fly, so this is, you will all recognize, a wing of a fly, and it's actually a very beautiful structure. It sort of comes out nearly perfect every time. And you can see, if I can get my pointer to work, here we go, that there are these lines on the wings, here and here and here. These are actually veins. It's a cross vein, and you get the same pattern in every single fly. Except when you put in certain types of mutations that rev up the MAP kinase pathway, including MEC mutations that underlie CFC, and you see on this one, you can see where the Roman numerals are like here, and here, and here, these extra lines. This isn't schmutz, this is actually extra wing veins that the fly body has made. So that, using that sort of approach, and there are other parts of the body that are affected well, he was able to learn several things so far about the MEC mutations we see in CFC. So first of all, he has discovered that the various ones have quite different strength in terms of how much they affect the fly's body. And he's been able to show, and this is really the more interesting part, that certain parts of the body get revved up by this gain of function mutation in MEC, but then there are other parts of the body where actually it gets turned down. the same gain of function. How that happens, we don't really understand completely, but that's starting to give us new insights to understand how different parts of the body are changing. So where are we going with this in the future? We need to know whether what we're figuring out in the fly is important or really relevant to human disease. So is it true that children with certain mutations in CFC, MEC mutations in this case, but he will go on to do other mutations like BRAF, is that true of the patients we see? Because so we can give parents more careful information when they have a baby about what's likely to happen. That's where the registry, by the way, becomes very important, because it will give us lots of information about lots of kids with CFC, and we'll be able to actually test that idea in patients. And then the hope is that some of these new insights that his work is giving us is going to inform the attempts to develop therapy. Okay, on to our little furry friends, the mice. So it turns out that when you put the mutation, in this case there are a couple models of BRAF that are mutations seen in CFC. And when you do that, you get features in the mouse that are like patients. So now I'm going to give a trigger alert. If anybody is like dreadfully afraid of mice, I'm about to show a picture of a mouse. So they're dead. They're not going to run across the carpet here. So here we go. On the top is a regular old wild-type mouse, a laboratory mouse. They're white, this particular strain of mouse. And you can see below the mouse that has CFC. And I think you can see very quickly that this mouse is quite a bit smaller than that mouse, even though they're the exact same age. In fact, they were of the same litter. And you can see that there are abnormalities in the way the spine formed in this particular mouse. And if you study the facial features, they're abnormal. And if you study the hearts of these mice, they're also abnormal. So shown here is the wild-type mouse pulmonary valve. So the leaflet is, you can see the thickness here. And you can appreciate here this sort of wide, blobby thing. It's a very abnormal pulmonary valve, so this is the mouse version of pulmonary valve stenosis, which I know that many of your children have that problem. So the mouse does a pretty good job of recapitulating the findings in CFC, and of course mice are a lot closer to humans biologically, so that's great. And so what are we actually learning from these mice? So first of all, we're learning that CFC looks different in the mouse as you have the genetics change in the mouse. So there are different strains of mouse, just people are different, and if you put the same CFC mutation on different mice, you get CFC differently. So we're starting to learn about that, and they're starting to test certain things that might ultimately use the patient in the mice to see if we can improve the outcomes in the mouse. So where are we going with this, or where are the investigators going with this? I'm not personally working with these mice, is to learn more about these abnormalities and how they form, like how do those heart defects form, like the pulmonary valve stenosis, to try a variety of therapies. And the thing that hasn't been done yet, that I think will be certainly coming, but it's very important, is to learn whether these mice show neurodevelopmental changes. We know that that is the case in other mouse models of RAS pathway disorders, like the Noonan syndrome mice have problems with learning and such, and that can be tested in these mice as well, and we'll see where we go with it. The other kind of modeling is the work that my group has been doing, so I want to tell you a little bit about that, and that has to do with the use of stem cells. So stem cells are cells that can make many other types of cells in the body, and pluripotent stem cells are ones that can make all parts of the body. So they can make the kidneys, they can make the heart, they can make the skin, they make the brain, and so on. And traditionally, we thought of stem cells as something, especially the pluripotent ones, that only Mother Nature could make. That was basically shortly after egg and sperm got together, you had a cell that could make anything that was pluripotent. And then we had a very remarkable discovery that resulted in a Nobel Prize by Shinya Yamanaka and others to show us that it was actually possible to take cells that were well past stem cell, that had already become something like a skin cell, and make time go backwards, to make the process go backwards. And that process is now called reprogramming, and this is being done all over the world in laboratories, including my own, and it allows us to take skin cells, or we can take blood cells, put in some magic potion, details not important, and presto magico, we get out stem cells. And these stem cells, these pluripotent stem cells, given the right recipe, can then be forced to become other types of cells. So you can take the stem cell and you can make neural cells, neurons. You can make heart muscle cells, you can make blood cells in the dish. as you want. So my group has been focusing mostly, we've done a little work with blood, but we've been, I'm a cardiologist, we've been focusing most on heart. And this is as close to science fiction, I think, as it gets. So here we've taken some of these stem cells and we have put in the recipe and they become heart cells. And as you can see, as you watch, these things are beating. They're not beating because we're giving them anything. They beat all by themselves just the way your heart does right now. So that's, just think about that for a moment. I can take a piece of skin from any of you in the room and make your heart cells and get them to work in a dish. So that's pretty amazing. And you might say, well, that's just a blob of cells moving around. Can you make something that actually, does some work? And the answer is yes, you can. So you can make heart tissue. So you can see here we've made a rubber band-like thing. It's sitting on 2 posts of silicon, and it pulls. And it just, again, we're not stimulating it. It does this all by itself. So that gives you the opportunities to study certain types of problems. And the problem that's easiest to study, actually, with these sort of approaches is hypertrophic cardiomyopathy. And indeed, we've done that. In the last year or so, we published the first model of CFC that was done in part with Kate's help because she had some of these stem cells. We had a line or 2 ourselves, and we put them all together. And we're able to study the features of hypertrophic cardiomyopathy related to CFC in a dish. And so here, just to give you some ideas of what I'm talking about, so shown on the left, these are heart muscle cells. There's the ones stained in green are heart muscle cells. And you can see that the size of a control or so-called wild-type human cell is very much smaller than the size of the cells we're getting from the made from an individual with CFC. So that's so that again we're able to get in a dish exactly what we would know that we're seeing in the patient. So then we're able to study this, and what have we learned? Well, we've learned that turns out the cells are talking to one another, and a large part of the hypertrophy or the overgrowth of the cell is from that conversation they're having and potentially interrupting that conversation as a way to treat this problem. And we do have the idea based on our work that this could potentially respond to drugs. So in the future, we have to see whether this is true for RASopathy, hypertrophic cardiomyopathy in general. And it's nice to do it in a dish with human cells. That's informative. It would be nice to try this in a mouse model and make hypertrophic cardiomyopathy in a mouse go away, and then maybe we could move on someday to training your children, if it was appropriate. Which brings me to the third part of the talk, which is about how are we going to go about coming up with therapies. And Kate alluded to this in her talk, and I want to go into this a little bit more. So the first question we have to ask ourselves is, if we had the right medicine for CFC, and we'll come back to what that might mean, which aspects could we potentially treat? And we don't really know, is the truth of the matter. I've made a list here. Some people I know would ask quickly whether GI problems could be on the list, and the answer is maybe. I don't know whether, if we had the right medicine and we gave it to a child with CFC, whether their GI problems would improve. And I don't know whether any of these problems actually would improve, but we do actually have some information on most all these problems to suggest that they would. And so they're really good targets. I put hypertrophic cardiomyopathy at the top because if you go talk to a drug company representative about this possibility, that's what they would put at the top. They don't like things that go after the brain. They've had so much failure in treating brain-related problems that they tend to be kind of negative about that. So it will take some work to convince them that that's a good target. But these are some ideas of where we could go. So, okay, so how are we going to find the drug? So basically, we have 2 possible ways of finding drugs to treat CFC. The first way is called repurposing. This is where you take drugs that the FDA has already approved that are being used for other things, hopefully very safely, I'll come back to that in a second, and then you repurpose them because you have reason to believe that they'll work for CFC. And Kate alluded to this, but there are many drugs being developed for cancer. Many of them are targeting the RAS pathway, and that's one area where we can think about repurposing. The other way to go about this is finding a new drug, something that would be specific for CFC and the other RASopathies. And I'll talk about the relative advantages and disadvantages for those. Okay, so what are the advantages of repurposing? Well, it's the fastest way to actually be able to help your kids. Because if it works, we have a drug that's already a drug, it already works, the FDA already knows about its safety or lack of safety, And hopefully, it's even been tried in kids. So that would be the best reason to try repurposing. The potential disadvantages has to do with the side effect profiles. And there we have the interesting conundrum, which is that we know that the drug companies are doing a lot to attack cancer. They know that 30 to 40% of certain cancers, particularly solid organ cancers in adults, cancers like breast cancer, lung cancer, bladder cancer, melanoma, all have mutations that occur in the cancer. The person wasn't born with them, they're acquired, they're not inherited. That also cause gain of function in the pathway, the RAS pathway, same pathway. And they know, the reason they're attacking it so hard is they know that if they can block the pathway, those cancer cells die. So that's why they've been working on it. There's already one very strong pathway inhibitor that's been developed, and that sits right here. It blocks MEK, and that has already been FDA approved for use in melanoma. But the question that we have to grapple with is whether the benefit of those potential medications for children with CFC is worth potential risk, because these medications have side effects. So here's how at least I think about the inhibitor. So some positives are, certainly we might be willing to try it in a life-threatening situation. So for example, there are some children, more Noonan than CFC, but in CFC occasionally, who have a form of hypertrophic cardiomyopathy that really is life-threatening. It's very fast, presents in infancy, and the children go into heart failure, and we know that those children don't tend to survive the first year unless something is done dramatically like a heart transplant. So that would be one group that might be potentially useful, and where we would be willing to tolerate a little bit of side effect to save their lives. And the other possibility that's theoretical, we just don't know, is that it could be that the side effects in kids with CFC would be a lot less than a person with cancer. Why? In cancer, the only place in the body where the RAS pathway has been zoomed up is in the cancer itself, right? This is an acquired mutation, it's only in the cancer. In kids with CFC, the gain of function is from head to toe. So it's possible that these drugs, which haven't been without side effects, might be better in kids with CFC. That's something we'd have to work through. Now, what are the negatives? Well, for many of the indications, let's take the neurodevelopmental indication. If we wanted your kids to be able to learn better, they're getting along. There are lots of challenges, but I don't think any of you or any of us on the physician scientist side would find any side effect virtually acceptable. Hurting your kid in any way in the attempt to make their learning better is just not an acceptable trade-off. So that's a high bar to get over, to have no side effects for these drugs that hit a very important biological pathway. Some people have thought, okay, we'll just give little bits of the drug. In general, that doesn't work with medicines. Giving less, you lose the side effect and you lose the helping effect at about the same rate. So that might not work. And then the other problem we have to confront in CFC, particularly I would say for things like seizures, neurodevelopment, is quite likely the drug will need to be taken for life. Remember, in cancer, they're just trying to knock out cancer. They'll give the drug for only a short period of time, relatively speaking. Here we're talking about potentially lifelong therapy. So this drug has to be perfect in order to make it work. So that's what we're up against. Now, an interesting alternative that we don't have yet, but just to think about, is there are drugs being developed that don't hit the normal protein. They only hit the protein that has the gain of function. And in that case, they leave the normal protein alone and typically have much lower side effects. And we know that there are such things that are being tested for SHA2, which is the PTP11 protein, and there's some new things like that being tried for RAS as well. Obviously, that might be relevant for KRAS and CFC. So if we could, for instance, get a drug that would hit only the activated form of BRAF, or only the activated forms of MEK1 or MEK2, those might be excellent things to try. At the moment, we don't have that, but that's one place to go. So now I just want to touch on the drug discovery part. My group has been doing a lot of the work in this area. The advantages of doing this is we're developing things that are specifically for the RAS alpha disease, not just CFC, but all of them, and we'll see where they would work. And the reason that might be different than what the drug companies are doing is because, as I told you, they just want to shut off the pathway to let the cancer cells die. We don't want to do that. We just want to calm things down a little bit and tone it down. And that's a little bit of a different task. So it's likely that there could be different drugs for that. What's the downside of the approach we're taking? It could take a long time. Somebody asked me in the room yesterday whether they thought we'd be alive to see it. I'm wondering about that in my career. I hope I will be. But there's no given. You know, it takes 10 years or more sometimes to develop this. It takes a long time. And success rate. You know, drug companies fail a heck of a lot when they start on these pathways, and we'll have to see. And we have to do this at the moment with fairly limited interest from industry. When you approach them about developing drugs specifically for CFC or Noonan or all of them, they're not like jumping up and down and saying, when do we get started? They're saying, let me know when you have it and we'll help you bring it to market. That's what they want. So we have to find funding from federal sources mostly, through NIH, in order to develop these drugs, in order to get far enough along to get the drug companies excited about the fact that this was something they should do. So in our work, we're doing it with fruit flies, a kind of crazy way to do it, but we have models, and we are testing both trying the repurposing approach using FDA drugs, and we're trying novel compounds. And at this point, from the novel compounds, we've actually identified 4 compounds that seem to have good activity, and we are moving those forward to try and make them into real drugs. We need to test them to see if we can block what I showed you in the heart muscle cells that we make from the pluripotent stem cells, and then we have to move on to the mouse and see if we can make the mouse better too. And then at some point we will be ready to move on to work on whether they're safe and effective in humans. Now, one question I want to touch on, because it almost always comes up, is everybody is hearing quite a lot about gene editing. This notion that you can fix the mutation in the cell and thereby treat patients in that way. And what has made that such an active conversation and brought it into everyday newspapers and magazines is, even if you read it online, I don't know if they actually reads it. Okay. Is the discovery of this system called CRISPR-Cas9. It comes from bacteria. There was just a huge patent fight about it. There will be a Nobel Prize for it sooner or later. But this is a system that the bacteria use that we've adopted in the lab to be able to edit the genome. So this is amazing. We've done this in my lab. Many, many labs all over the world are doing this. You can take a cell and you can create the mutation of CFC. You can take a cell that has the mutation of CFC and you can turn it back to not having the mutation of CFC. So this is an amazing tool for scientists. I do believe it will be useful for therapy. The therapy it will be used for first, almost certainly, are for diseases where you can take out a sample from of blood or bone marrow, take the cells, stem cells from the bone marrow, fix the mutation, put it back in the bone marrow, have the cells then, being divided in the bone marrow, and that will be the treatment. So that would be really good for certain types of diseases. So for instance, sickle cell disease would be a good example. That's something that's a blood problem per se. The mutation is in the cells, of course. If you could fix those blood stem cells and then put back the fixed ones, you'd be good to go. That's going to work well. That is not going to work anytime soon, possibly ever, for something like CFC. Because think about it. We would have to find a way to fix the mutation in high percentages of cells in several organs. Let's just take the brain. Just getting this in there and fixing all these neurons, and typically the effectiveness so far, this is very, very, it's the rare cell that gets fixed. And when you do it in a dish, you can pick out the one that's fixed and put that one back. But in the person, so far we're nowhere close. So I don't see this approach being useful for CFC or the other RASopathies, where it's gain of function, we really have to fix the mutation to do it. I don't see that happening anytime soon. Don't mean to be negative, but I know there's so much excitement about that, so I just want to put that out there. And if people have questions, you can ask me that when I'm done, which is in just a few moments. Now, I want to mention, the last piece is I want to mention about this effort that's going on called CleanGen. And the leadership of your organization is having discussions, and of course people like Kate, myself, and others are interacting with them to help decide whether this is something to participate in. But I want to at least mention it so it's familiar, because I'm hopeful that we will get to a point where it's something we will be able to do with your registry to help your community and help other communities. So the problem we have right now is broadly speaking, is there are many, many genes for many, many genetic traits. CFC is one of them, as you know, there are the 4 genes, but there are, and then you move outside the RASopathy, there are at this point probably 1,000 or more genes. And there's genetic testing being done, and there are lots of laboratories all over the country, and in fact in other countries as well. The problem is there isn't really agreement on what a mutation is necessarily. And each lab is sort of doing their own thing. There's no centralized way, and there's no sort of jury sitting and saying, that's a mutation, that's not a mutation, I'm not sure about that one. And the idea is to try and bring things together so we get some agreement to say, yes, this is a gene that causes disease or diseases. These are the changes in that gene that we are sure cause disease and so on. And to put that in one place and bring that all together. So that's what ClinGen is trying to do. And to do that, they've been funded through the government by the National Institutes of Health. It's an important effort. And the whole idea is to bring together experts to interpret these things and then come to common agreement about them. So, and it turns out that each disease is like its own little country. You have to make decisions about what's a gene and what's a mutation for that. So they've started with these groups, and you'll be pleased to see that down here are the RASopathies. So one of the groups that they put together from the get-go, I don't know how these were picked, I had nothing to do with it, were the RASopathies. And then they came looking for people who could serve to be on their panel for that. And you will see a couple of familiar people, hopefully. Amy Roberts. Amy's not here this year, but she's been involved with you guys. I'm here, obviously. Kate's involved. And then there are a number of others involved. Some of them are on the lab side, some of them are counselors, and so on and so forth. And we've been working together for a while and coming up with rules and way to do it. And again, the idea is to then take data and share it and have it be publicly available and hopefully get the labs unified about what they do, and it'll also be a good research tool. So they are, of course, looking for information about patients, both their clinical testing from labs and also about the patients themselves. So there are 3 ways, basically, that this can be done. There's the possibility that individual patients could do it, but that's kind of a pain in the neck, and for your group that makes no sense because we've just heard you're developing your own registry and nobody wants to have to. Doing it once is very important. Being asked to do it a second time, not such a good idea. So we're not keen on that. But what we are keen on is allowing your registry, as it builds out, to provide its data, again, in an anonymous fashion, so nobody will know who's who, to this ClinGen effort, so we can have it together. We'll bring it together. Noonan syndrome is doing the same. We're hopeful that Costello syndrome will do the same, and we'll have all of them residing in a single place. And that will mean that when the next family has a kid born with a RASopathy, and the testing is done at whatever lab, the ability of that lab to be sure about what's a mutation or not will improve. And then hopefully, as we learn more about individual mutations, we'll be able to say something better to parents to give them advice what to look out for in their kids as well as for the doctors caring for those kids. So that's the idea behind ClinGen, and we're pretty lucky because the platform, the computer-based platform that ClinGen is working on is working with Invitae and is completely, term for it, but operational with the registry that you have put together and that will be expanding hopefully in the weeks and months to come. So I think it's a really great opportunity. It will result in helping a lot of other people, and it would be a really good early test case for the use of your own registry. So I hope we'll get there. So I'm going to stop there. I'd be happy, I'd have some time, and I'll be happy to answer any questions that you have. Thank you very much.
We have time for a few questions. Can you raise their hand?
Thank you. Can research from other RASopathies be applied to CFC?
Yes and no. So it is the case that there's research going on for Noonan syndrome, there's research going on for Costello. They are, with rare exception, all gain of function in the RAS pathway. Our hope would be that what we learn about one will be be applicable to another, that a treatment that works for one would work for many others, hopefully all of the others. On the other hand, that may not prove to be the case. We'll have to wait and see. We know already that, for instance, when we make, I have a project going with a collaborator at Mount Sinai, also with flies, where we put in 20 different mutations, RAS-optin mutations, some for CFC, some and so on. And quite interestingly, the effects are really different on the fly. Some of them have abnormal wings, the way I showed you. Some of them have abnormal eyes. Some of them have an abnormal back. Some of them have multiple. And so they're not all acting exactly the same. So we're going to have to, but that's actually a strength. By studying those various separate ones, we'll learn more about all of them. So that's actually helpful, I think, for the CFC community, because obviously the aggregate of RAS-opathies are commoner than just CFC alone. Same could be said for any of them, and of course Noonan is the commonest. So, and hopefully a therapy, if it worked, would be more attractive for industry if it could work for multiple genes and multiple traits. But time will tell.
We have time for one more final question.
Hi, you have mentioned that the mutation sometimes has the neurons go up or the neurons go down. Is there any way to have like a drug that could, make it, when it's going up to level it off, and when it's going down to level it off as well?
Right. So that's a really good question. So the issue we believe is, the effect of the mutation is always to drive the protein up. But built into, we always show the pathway as I showed it, as Kate showed it, as this beautiful domino thing, which is complete fiction, actually. There are lots of other loops and interactions and stuff that we don't begin to understand, and the hope would be that if we can find something that sort of nudges the pathway towards normal, that those regulatory things that are causing it to turn down in some tissues will themselves get turned down, and therefore the signaling will come up to our normal level. So the hope would be that, if we find the right agent, and the way we're going about it is actually agnostic, meaning we don't care which way it goes, to whether ERK is up or down. We just want the fly to be normal, and then if the fly's normal, then we'll move on to whether we can make people improve with it. That's been our approach. We'll see if it works. Okay. Thank you very much.